for that. The next one was looking at transcription and translation. It's fun, right? So everybody knows this stepwise process, going from a DNA gene leading to the mRNA transcript, and then it leads to protein production. Make sure you know proteins are the component that involves the phenotype of the cell. The transcription process involves RNA. Okay, so RNA, as we talked about, there are 3 distinctive functional types. You have messenger RNA and RNA, ribosomal RNA or rRNA, and then transfer RNA or tRNA. So all of those, again, in relation to what they do for transcription translation. The other thing that was important that's different from DNA is what? Thymine is switched out for another nucleotide known as uracil. So remember, you would expect to see thymine on the RNA strand is going to be replaced with uracil. So if you see U in a strand like this, you should know that this is RNA and not DNA, correct?
Yeah.
Okay. All right. Base pairing rules, all that stuff we talked about. So transcription, make sure you know the enzyme that's involved in this. Okay, transcription, the involvement of the enzyme is RNA polymerase. Make sure you know down here at the bottom, so when a termination occurs, what happens primarily is that hairpin loop that's formed that causes transcription to be terminated. Translation is associated with what? The ribosome. Okay. Make sure you know what the starting codon sequence is and what amino acid associates with the starting codon in the protein chain, which is FMET. Does everybody see that right there? Okay, ribosome. Okay, starting codon, FMET. All right. The other part of this was in relation to how the ribosome moves down the RNA, which is down here in the 3rd bullet point, which is translocation, moving down the mRNA, going from 5' to 3'. Okay, so 5' to 3', and it's going how many bases at a time? 3 bases, known as a what? Codon. Okay, so everybody remember that? Codon has how many bases? 3 bases. Okay, start codon, like you said, a UG. Okay, and again with the translation location of the ribosome down the length of the mRNA. I'm not going to ask anything on termination on this, so I think that's it for that one. I don't think I'm going to ask anything on this one. Genetic therapy. The only thing that I may ask on this one is the CRISPR-Cas9 system right here. Make sure you know what this is right here that's listed in relation to genetic therapy. So this is the one that can take out mutated strands or genes and replace them with the correct one. So this is technology that's helped to treat genetic therapies or genetic diseases. One of these includes HIV resistance. Okay, so this is an important aspect related to T cells that express this protein receptor, which is right there, right? So CCR5. So if you have a mutated CCR5 receptor, that makes you resistant to HIV infections in your T cells. Okay. So what they did with this was they were taking somebody that was HIV infected, reprogramming their cells to generate this mutated CCR5 receptor that makes them resistant to HIV. So I think that's it. As far as this one is concerned, antibiotics, make sure you know the man that's, our person that's responsible for discovering penicillin, which is Alexander Fleming. Okay. I think I'm going to ask you anything on that. Now, as far as resistance mechanisms are concerned, we talked about 4 different types of resistance mechanisms that are associated with bacteria. These are listed here on the next 2 pages: production of drug inactivating enzymes, modification of a target molecule, decreased influx or uptake of the bacteria, and then increased elimination of the internalized drug. This is summarized on this page right here, so that's all of those right there, so make sure you review those, okay? Make sure you know those. We did talk about the CDC report in relation to the different types of prioritized categories. There are 3 different ones they listed in this 2019 report, which included the urgent hazard level threat, which is the high consequence associated to public health related to drug resistance, serious hazard level, which is associated with a significant amount of antibiotic resistance related to affecting public health. And the last one is concerning hazard level, which is associated again with a lowered amount of drug resistance affecting public health. And I think that's it on this one.
I think in the exam, I might be mistaken, you asked about which one has a higher risk and then which one has a lower risk. Those were 2 different questions. That would be the first one, which is urgent, and then concerning, right?
Right, so if you list them out, so urgent is the highest, right? So just make sure you know the ranges associated with that defined list. So urgent is the highest, and of course, related to the rest of the ones that are listed. You don't have to remember the organisms or anything, just in relation to the effects to public health, yeah. Okay, any other questions? Okay. Take home quiz number 2. So we only have 1 more to go over here. So viruses. So this one, I do want you guys to go back and review that T4. So everybody remember T4, the steps associated with T4. Make sure you go back and review these in order in relation to how T4 replicates in E. coli. Okay? Any questions about that? Okay. Viruses part 2. This is in relation to eukaryotic viruses and how they infect humans. One of the things we talked about were viral families down here that viruses can be grouped into based on genomic structure, shape of the capsid, and then the presence or absence of an envelope. We did talk about 5 different families that are listed here. We went over each one of those in relation to the characteristics of the genomic characteristics, the shape, presence or absence of an envelope. The other thing that we did talk about with these organisms or viruses is that how they are released by the cell, okay, which is right here. For Coronaviridae, for example, they're released by lysing the cell. Retroviridae released by budding from the cell. Denoviridae released by lysing the cell, and then Filoviridae released by budding. Now, the other family we looked at were the coronaviruses. Okay, so the coronaviruses, we talked about how we have these different types of viruses that are up here that are associated with humans. One of the primary targeting receptors that are associated with coronaviruses in humans, in particular the SARS-CoV-2 virus associated with COVID-19, is this one right here known as the angiotensin converting enzyme 2 or ACE2 receptor. Okay, so make sure you guys know that as the primary receptor for this particular organism. The other thing is that the budding mechanism for this organism is different, right? So for this particular virus, it uses this ER Golgi intermediate compartment to actually bud into and then be released from the cell by exocytosis. So make sure you know that SARS-CoV-2 is the cause of COVID-19. Everybody should know that by now, has led to the epidemic that we are currently in. I think that is, I may ask you a question here on the Spanish flu as well. Knights flu of 1918. So making sure that you remember that this particular flu associated with an age range of 20 to 40 years of age primarily that were mostly susceptible to this, and that the type of virus that they were able to identify back in 2007 was H1N1 influenza type A virus, which is listed here. I'm not going to ask anything else on this. Non-specific immunity. 3 essential functions of the immune response: recognition, containment, clearance. We've talked about that over and over and over again. Okay, so with the specific innate immune cells that we talked about included neutrophils. Okay, so make sure you know what neutrophils are. And then down here, make sure you know what dendritic and macrophages are, as they're listed down here. Okay, the bottom. The other part of this was knowing the steps to phagocytosis. So you guys remember all that phagocytosis steps? Okay. I think that's it from that one. Now we're at specific immunity. So specific immunity, we talked about the B and the T cells. Down here where it says T cell mediated or cell mediated immunity is associated with the T cells, and then humoral mediated or antibody mediated immunity is associated with the B cell population, right? So T cells, cell mediated immunity, B cells, humoral or antibody mediated immunity. The other things that we talked about in relation to T cells were presenting of antigens, right? So does everybody understand the differences between MHC1 and MHC2? So make sure you guys review this, okay, in relation to where they're found at, what types of antigens they present, and what T cells recognize antigens that are presented on this. The last part of this was going back to review your antibody classes. So you have IgG, okay, IgM, IgA, IgE, and IgD. So the ones that I'm really focusing in on for the test would be IgE, IgM, and IgA. So those are the 3 I want you guys to review over for the test. Okay, the rest of them don't worry about. Right, so IgG, IgM, and IgA, right, in relation to infections. Any questions about that one?
Just to clarify, you said IgG, not IgE.
IgG, IgM, and IgA. Yeah. Okay, so let's see. Make sure you know what propagated spread is. Okay, so propagated spread in relation to how disease or infections are spread within the population. And then the other thing was down here is community-acquired versus hospital-acquired infections. So just review those. Make sure you know the definitions related to how it develops. If it's associated with obviously a hospital-acquired infection, if you get it after being admitted to the exempted of hospital administration, then that's not going to necessarily be hospital-acquired, but community-acquired infection. All right, so I think that's it, right? So that's what we had for the other tests. So again, I will not be here on Thursday, so no class on Thursday. Let me know if you have any questions or anything like that. We have lecture exam number 4 a week from tonight in this class. So